From Microbe TV, this is TWiP, This Week in Parasitism, episode 184, recorded on June 4th, 2020. I'm Vincent Racaniello, and joining me today from Fort Lee, New Jersey, Dickson Despommier.
Hello there, Vincent.
How have you been? Fishing?
Well, thank you. Yeah, I went fishing a few times. Took my grandson out to the fishing scene that I ordinarily go to, and had a great time.
All right. Also joining us from New York State, Daniel Griffin.
Hello, everyone.
Hey, Daniel.
Have you been fishing?
You know, I haven't. I've been working too much.
I know you have.
I got to stop this.
Daniel, let me just ask you a quick COVID question.
Okay.
Can you see the day when you have zero COVID patients in the hospital?
I actually think we might be getting pretty close. We had a patient today who came in April 15th who just got discharged, did not die, discharged to Florida.
Wow.
We're down to 3 active COVID patients. So these are people who are active. But the interesting thing is, am I ever going to see no COVID? We're seeing a lot of late stage. These are people who had COVID 4 weeks ago, and now they have Staph aureus in the blood, E. coli in the blood. We actually had a gentleman who had been in and out of the hospital, urgent cares, the ER, came in with a nice right lower lobe pneumonia, but these fevers every day of 103, it didn't make any sense. We did COVID serology, and he's got COVID, positive IgG. So we're seeing this late COVID effect, but I think we're probably going to get to the point where maybe for a little window here, we actually have no acute COVIDs. I'm hoping.
So Daniel, what was your maximum load at one point?
We were up to, at the one hospital, it was just one of my partners, Dr. Lee, and I, we were up to over 150 active COVID patients at one time. And for a long time, we were basically about 130 was our average for a couple months there.
How's the Irishman doing?
So the Irishman, I think as I mentioned, he went home. Well, didn't go home. He went to a rehabilitation center. I mean, being sick that long took something out of him. He was briefly readmitted to the hospital with bacteremia, actually an extended spectrum beta-lactamase, very resistant bacteria in the blood that we think was from a urinary tract infection. Sort of late stage. He's now back in rehab, getting his strength back, and hopefully at some point we're going to get him back out to Colorado.
Wow.
How old a guy is he?
He's in his 30s.
Oh, wow. That's not old at all.
No, not at all.
No.
All right. Well, this is TWiP, so Daniel, that means case history or case. Tell us about the last case.
Yes. So usually you've got like a nice summary for me.
It's there.
So let me go. Let me, as I like to say, let me go through the case summary. This is for those of you who are tuning in for the first time, as well as those who are tuning back in. And this was the case of a 70-year-old male who had returned with his wife from safari in South Africa. So this was going to be the safari of a lifetime, but he returns suffering from 2 weeks of watery diarrhea. When he went for this very fancy safari, there was a severe water shortage in South Africa. There were issues. There was limited hand washing. They were having them use hand sanitizer to clean their hands. So there was obviously an issue with hand hygiene all around. 5 days after he arrived, he and his wife developed watery diarrhea. And this was a lot of watery diarrhea. There was about 12 episodes a day. He felt nauseated, abdominal cramping. Then we actually went ahead and we got some information, stool culture. I mentioned that the O&P was negative. We did give some background. He was healthy with no significant past medical surgical, nothing running in the family. And then I said that we got a little more information. Stool was sent off for an acid fast stain, so a special oven parasite with acid-fast stain. We also ordered a GI PCR panel, and the results prompted me to treat him with 7 days of trimethoprim sulfamethoxazole, or Bactrim or Septra, and he got all better.
Cool.
All right. We have some listeners weighing in. Dickson, you want to take that first one?
I would love to. Josh writes, "Hi, TWiP hosts. I'd like to thank you for all keeping us updated in the world of parasitology in these busy times. I'm excited to answer this case study as coming from South Africa myself. I believe the safari goers have cyclosporiasis, caused most likely by ingesting contaminated food or drinking water containing sporulated oocysts of Cyclospora cayetanensis. My bet would be that there's a food handler is not engaging in good public hygiene practices. The giveaways were frequency of the watery diarrhea, typically 5 to 15 bowel movements a day, and Dr. Griffin's emphasis on trimethoprim sulfamethoxazole, which is the first drug of choice. I'm assuming the GI PCR panel came back positive for Cyclospora cayetanensis, and the good old Zeal-Nielsen stain microscopy revealed the oocysts looking like this, and then he submits a very nice picture, which could substitute for the one we have in our textbook of the causative agent that he thinks is causing this problem. He says, I'm hoping to go 3 for 3 in the latest case studies. Interestingly, the animal conservation and protection heavily relies on the tourist trade. The impact of COVID-19 is leading to a lack of revenue and funding for conservation efforts. Are we going to see an increase in poaching as nearby communities turn to it to obtain money in such harsh economic climate? In order to help, they have recently implemented virtual safaris. Oh, wow. I'm sure Dickson would like to enjoy a safari from the comfort of his living room at the fraction of a price. On a side note, thank you, Vincent, for the virology lectures on YouTube, and one lecture, number 25, sparked my interest recently. You went on to explain prions, or prions, depending on how you pronounce that, and mentioned exotic ungulate encephalopathy can be found in kudu. This sent me off to try to find out more about it. What interests me is that in South Africa, we don't seem to have any surveillance for it. I could be wrong. Many game farms have kudu, and they are bred as livestock as well. We also make biltong, which you refer to as jerky, with kudu. Would this be any worry for or indicate a need for surveillance? Are there any wild-type prions, exotic ungulate encephalopathies out there being transmitted across kudus, similarly to the deer wasting disease crisis in the U.S.? Sorry to go off on track. Thanks again for the TWiP. Thanks again, TWiP hosts, from Josh from Cape Town, South Africa. Yeah, Vincent, what do you think about that? I haven't heard anything about it. I was there, of course, probably at the same time this other person was there also, but we didn't hear anything about it.
There are, the kudu transmitted to each other. That's how it's spread, but it's pretty rare, so I don't think they're worried about getting into people. Although the jerky you're talking about would not be clear.
Safe.
It would not be safe, no.
I'm thinking a kudu that's exhibiting wild, crazy motility would be a perfect target for a lion or a leopard. So that might go to that group of animals too, you know?
Yeah, could be. Well, they're known, the great cats are known to have it as well.
Wow.
Mainly from zoos where we feed them contaminated meat.
Goodness.
But otherwise, it's very interesting.
Oh, yeah.
Daniel, I think you should take the next one.
Dan writes, not me, someone on different Dan,
right.
Just to clarify. I'm back for my second guess using the MO of control F PD7 for trimethoprim sulfamethoxazole brought me to Cyclospora cayetanensis as the most likely culprit. Symptoms appear a week or two after infection, 5 days a little short, and include 5 to 15 watery bowel movements per day. Sensitivity of microscopic identification of the oocysts can be improved with acid-fast staining, which could explain why the first O and P without it came back negative. Thanks for everything you guys do. You are all the heroes, Dan.
Heather writes, hello again, my micro friends. Well, you aren't small, but our shared interest in tiny pathogens is. I think the gentleman who traveled to South Africa has a cyclospora infection, according to CDC. Symptomatic patients may not shed enough oocytes in their stool to test positive for the standard stool culture O&P, requiring the acid-fast staining and PCR. My central deciding clue was that the Zithro didn't help him, but Bactrim did. I suspect he got it from produce he ate on his fancy package deal trip, since he was good about using bottled water. Mm-hmm. I'm a frequent hiker and camper in the Blue Ridge Smoky Mountains, so I'm familiar with Giardia, but the Zithro would have knocked that out if I'm not mistaken, and I don't think the first round of doctors would have missed that common hiker's foe. I hope I'm right. I just discovered TWiP in the last month, and I'm less familiar with parasitic infections than other maladies, but if I'm not right, don't worry. I'm not a doctor. By the way, this morning when you read my pinworm letter first, you made my week.
All right.
You guys are my close second favorite public figures at the moment, second only to Dr. Fauci, of course.
We're not okay.
My friends and family might be tired of me telling them what Dr. Daniel says is improving outcomes in COVID-19 treatment, but that's not going to stop me even once. Stay well from your favorite high school assistant principal.
Oh, that's cool.
You know, Dr. Fauci has kind of disappeared lately, hasn't
you're right.
he?
You're right. Absolutely.
Yeah, no, that is true. And he seems to be saying very little. I think
he said a lot already.
there may be some dynamic going on there.
There might be. We want to,
but I should comment. I noticed you cringed a little. I don't know if the listeners could hear your cringing, Dickson. But about the zithromycin.
Yeah, right. I cringed. I did cringe. I have to say.
Yeah. The azithromycin is not going to do anything about Giardia. No, just going to point that out.
Right.
Dickson,
Anthony writes, I am writing in once again to provide my guess for TWiP case number 183. The initial symptoms had made me think of crypto, but the long duration and abdominal cramping coupled with the interesting find on PCR panel immediately caused my mind to jump to cystoisospora belli, a rather rare gastrointestinal infection, but could perhaps be acquired in an area with rather poor sanitation. I think the fact that the patient recovered after a week of Bactrim really clinched the diagnosis for me. Keep up the amazing content, Anthony.
All right.
Well, thank you for your guess.
Elise writes, Dear TWiP trifecta, I do hope this finds all of you well. I was so pleased to hear that Dr. Racaniello remembered me, and I'm even more pleased to be writing back with a diagnosis guess for part one of the two-part case study presented in TWiP 183. I suspect the gentleman who went on safari in South Africa has been infected with cyclospora. The parasite is easily contracted via the fecal-oral route, and given that the patient and his wife were traveling in circumstances where washing was unreliable, it sounds like the stage was perfectly set for an infection. The symptom of frequent episodes of watery diarrhea, cramps, and nausea is common to a lot of parasitic infections, and I entertained a few other possible parasitic prime suspects like Giardia, but the symptoms are somewhat different. The diarrhea in Giardia cases is fatty and greasy versus the watery variety that the patient experienced. Cyclospora seems like a more likely parasitic culprit for several reasons. First, the gentleman's symptoms are most consistent. Secondly, he responded entirely favorably to the trimethoprim sulfamethoxazole treatment, which is a standard recommendation for treating cyclospora. And finally, his initial stool culture was negative. This is not uncommon with cyclospora infections, and one recommendation is to do multiple stool tests over a number of days, or alternatively, doing the additional tests that Dr. Griffin's patient had done, O&P acid fast stain, GI PCR panel. I'm now extremely intrigued to see how the patient's wife managed to get herself an entirely different parasite. I appreciate this case because I have been traveling virtually and watching these daily safaris in South Africa on Wild Earth. Here is a link if you too want to safari vicariously with no risk of parasites at midnight or 9:30 in the morning Eastern Standard Time. And we get a link here to wildearth.tv /Safari Live. It's sunny and about 63 degrees, 17 C, today in Lower Manhattan. Again, I'm so grateful to all of you. Big best wishes, Elise.
Cool.
Welcome back. Glad to have two in a row.
Indeed.
Sophia writes, Dear dedicated doctors, as per the Italian dottore. I hope I make Daniel proud this time. I guess he really wants us to learn this stuff. Ancient Greeks said repetition is the mother of knowledge. So I will say the symptoms are due to Cyclospora cayetanensis. Of course, I cheated by looking at other TWiPs and Kevin's notes, who already has an A on this course and is just auditing for fun. I don't think I will ever remember this organism. Anyhow, I do have a question. If you're out traveling and you get infected with Cyclospora, will the symptoms eventually resolve without any treatment? Or is it the case that you really need medication to get rid of this? I usually take Imodium pills with me to stop the diarrhea, but this won't stop the organism from multiplying. Thank you for making time for TWiP, and happy birthday to Dickson.
Well, thank you.
Sophia, Greece 35C due to sandstorms from the Sahara.
Goodness.
Unusual for this time of year, but what's not unusual this year?
She's so right.
Indeed.
Wow.
Dickson.
Alexander writes, Dear professors, it's 24C75F out today, and it's just now starting to feel like early summer is just around the corner. I'm a med student in Vienna, Austria, currently working as a study coordinator for a COVID-19 trial. I've lately been skipping all of the podcasts in my subscription list that even reference that damn virus, with you and Christian Drustin's works being the only exception. This has given me the opportunity to explore all of your other series, and having only been a regular listener of TWiV and TWIM, discovering TWiP was a special treat in this situation. Over the last couple of days, I've binged your most recent episodes, and listening to your case of the safari tourist immediately rang a bell for me. The water shortage in the area of the resort they were staying in might be a prime cause here. While they felt they were safe drinking bottled water only, the kitchen might have skipped the washing of fresh produce, resulting in the transmission of the pathogens in question. The high frequency of diarrhea, the lack of fever, blood or steatorrhea, and the response of trimethoprim sulfamethoxazole all reminded me of the 2 volunteers in Uganda a few episodes back. This, of course, makes infection with Cyclosporidium cayetanensis the most likely diagnosis. This pesky fiend is endemic in South Africa, where a large portion of the vulnerable population is also at risk for complicated diseases due to high prevalence of HIV infection. Isospora belli, or cystoisospora belli as we now call it, might cause similar symptoms, but has a longer incubation period of 7 to 10 days and requires a more prolonged therapy. Blastocystis hominis may also be the culprit, but its role in pathogenesis remains controversial. Anyways, seeing as I have no experience in the field of parasitology except for listening to your podcasts, I might be the victim of the famous Dunning-Kruger effect here. I really enjoy everything you produce at Microbe TV. These stressful times, they're to be cherished reminder of what I want to do as a doctor what I'm passionate about. I'm looking forward to all future episodes of TWiP and the other TWiZs, and I hope you're all staying healthy and looking out for yourselves. Best regards, Alexander from Vienna.
I forgot to ask you, Daniel, in the previous email, she had said, do you have to treat this diarrhea, or does it self-resolve?
Right.
I guess this is going to be the issue, like whether or not it's Cyclospora cayetanensis or not. We'll have to see. If it happens to be Cyclospora cayetanensis, then I'll address that. If it's not, I'll address that as well.
Wow, he's a cagey guy, isn't he, Dickson?
He's heading towards politics.
Daniel, you're next.
Dear doctors, I think your 70-year-old patient returning from South Africa has Cyclospora cayetanensis. His frequent diarrhea, nausea, and abdominal cramping are consistent with the symptoms. The fact that he was staying in an area with severe water shortage, I got to work on the T. I slip into I was born in New York, so I tend to slip into severe water.
That's right.
The water shortage made good hand hygiene almost impossible. Another factor is that C. cayetanensis is endemic in many parts of the world, making it easier to become infected from contaminated food and water. I did consider the cause of his diarrhea could be from Cystoisospora belli because it is also diagnosed with a modified acid-fast stain and treated with trimethoprim sulfamethoxazole. However, I ruled out C. belli because there was no mention of fever in the patient, and C. belli infection is much less common. Thanks again for your great podcast. Sincerely, Cecilia, St. Petersburg, Florida.
Okay, Kush and Karishma write, Dear Trypanosomes, I'm writing into you from Birmingham, England, where it's currently 19°C and sunny with a cool breeze. My wife and I are both physicians in training and have been listeners to your fantastic podcast for a few years now, but have only just mustered up the courage to email in our thoughts on the case study. The clinical presentation of watery diarrhea in returning travelers, of course, has a very wide differential, so it was the need for an acid-fast stain and treatment with co-trimoxazole that narrowed it down. As far as I know, the acid-fast stain is used on stool to identify either Cryptosporidium, Cyclospora, or Cystoisospora. All of these can be a cause of watery diarrhea, but Cryptosporidium is treated with nitazoxanide or in HIV patients with heart, whereas cotrimoxazole is an effective treatment for both Cyclospora and Cystoisospora. As the diarrhea has been going on for 2 weeks in an immunocompetent individual, we reckon the most likely diagnosis here will be Cyclospora. Thank you for the awesome podcast. Keep up the good work.
James writes, my wife and I are going on a TWiP soon to see our daughters, grands and son-in-law in Tennessee. It is freaking arthropod central down here. Ticks, chiggers, skeeters, you name it. Anyway, 70-year-old dude goes to South Africa and gets watery diarrhea for 2 weeks. A bit long for E. coli or most viruses, and after all, this is TWiP. Regular O and P is negative, but acid-fast O and P is positive. This is a modification of the acid-fast stain used for mycobacteria, and it stains the oocysts of the big 3 C's, Cryptosporidium, Cyclospora, and Cystoisospora. Used to be Isospora, as I recall. The oocysts of Cryptosporidium are somewhat smaller than those of the other two. My medical students actually knew that a couple of weeks ago from their parasite quiz. Oh, good for them. And also are susceptible to Septra, Bactrim, trimethoprim sulfa. I'm not certain that we have enough to distinguish between C. belli and C. cayetanensis, but who cares? My PhD cares, but my MD doesn't care. I did diagnose a few of these coccidians of small bowel biopsies back in my active pathology days. Oh, you'll like this. I also diagnosed 2 cases of Whipples, and we even did an EM on the biopsy. The San Francisco HIV site says that Cryptosporidium is 4 to 6 microns and about the size of a Canada yeast. Isospora is about twice that size, and Cystoisospora is up to 25 microns. But I'm sure there are better molecular tests out there in countries that can afford them. Teaching is interesting with Zoom, recorded PowerPoints, emails, and message boards, but so far so good. Got excellent reviews for the micro course this year. Taught from my stand-up desk in the corner of my home office. It's looking like most of my work will be done here at least until January and possibly next June. TWiP on, dudes. James writes, he's got an MD, a PhD, and an FCAP, which is a board certification in pathology. That's great. Glad you're looking at us and listening to us.
Daniel, you're next.
Yes. Andrew writes, Kia ora to Grumpy, Sleepy, and Doc from Pongaroa. I think he must listen to TWiP also.
Absolutely.
In my opinion, Dickson should be called Happy because he tells the best joke.
Thank you.
And it is his choice. No book one yet, but that is okay. I think he's sort of trying to get you to send him a book. I'm secretly waiting for the 8th edition. Weather is 16°C and raining. COVID-19, we are now in level 2, and we have just 27 active cases, down from 929, and countrywide travel is now allowed, but social gatherings are limited to 100 people, with exceptions for some funerals due to cultural factors. Schools are open, as are restaurants and bars. These restrictions led to our prime minister and her family being carried across to a popular cafe. The happy end was that a table freed up and a staff member raced down the road to inform her she could come in. Public transport and shops are required to maintain the 2-meter social distancing, but masks are not required. The borders remain closed to all non-citizens. There is talk of establishing open borders with other countries that have or will soon achieve zero community transmission. This will be important for our tourist industry and, curiously, people waiting for overseas post. Many countries cannot send us mail to the lack of aircraft flying to and from our shores. One of many third-order consequences of the pandemic. The ramifications of the pandemic will last well into the coming decades, and I hope that we will use mountains of data collected to prepare for the next pandemic, whatever the pathogen is, viral, bacterial, or parasitic. My guess for the man who holidayed in a drought-stricken South Africa was suffering from diarrhea. As I see it, there is not enough information to determine which parasite was in play, even if I rule out those relying on insect vectors because of the dry conditions. I'm thinking the wife's symptoms might make it clear which organism was responsible, and that being the reason Daniel has a sequel to this case, TWiP 184. I made a table and checked off those parasites left against the symptoms, geographic spread, transmission route, tests and treatment, the idea being to allocate a probability to each. The surprising factor for me that limited the selection the most was the successful treatment with TMP sulfa. So my Bayesian priors are Cyclospora cayetanensis, 55%, Cystoisospora belli, 25%, Blastocystis hominis, 10%, something else, 10%. I am really looking forward to the denouement. And this is Naga Michi from Andrew.
Jim writes, greetings, distinguished antagonists of the viral misinformation. Sometimes people who travel abroad to see exotic wildlife will encounter more creatures than meet the eye. In this case, the two patients were probably infected via the fecal-oral highway with the parasite Cyclospora, which they might have contracted while eating at the lodge. The water shortage could have adversely affected normal standards of uncooked food preparation in addition to fostering poor hand hygiene. As we learned in an earlier TWiP released around the dawn of the COVID-19 era, the medication known as Bactrim should be effective against this protozoan parasite. Many thanks to the three of you for your continued leadership on TWiP and TWiV, and especially to Vincent for all the other podcasts in the Twix universe. You guys are making a real difference. I would urge all those who have not yet contributed to add their support through Patreon. Take care, be safe, and remain humble. TWiP rocks the roiling 20s. Wonderful. Wonderful.
Bethany writes, hello. I'm a relatively new listener and first-time case study respondent. I was a few episodes behind because I've been trying to keep up on all the wonderful TWiV episodes released, but I finally binge-listened to a bunch today. Lucky for me, I think that the diagnosis for this case is the same as episode 181, Cyclospora cayetanensis. Over 10 episodes of watery diarrhea and resolution after Bactrim treatment were the main clues for me. I don't know if this email is too late or if you read all of the emails you receive, but as a PhD student in infectious diseases, I would love to expand my infectious disease textbook repertoire. Thank you for all you do. Fauci for president! Regards, Bethany. I second the motion.
All right. University of Central Lancashire Parasitology Club writes, Dear TWiP professors, hello again from the Parasitology Club at the University of Central Lancashire in the beautiful northwest of England. This strange semester is more or less over having switched to online teaching and examinations in March due to COVID-19 restrictions. Marking of assignments and exams has been completed, so the summer has started for students and planning for next semester has started for staff, and we have beautiful weather to cheer our spirits further. The case of the gentleman of 70 years and 10 returning with his wife from South Africa with more water in his stool than was available for hand washing on his African sojourn. I like that. The profuse diarrhea, nausea, and abdominal cramping would suggest enteritis, and without the narrowing of the field to parasitology, would suggest an infection with a toxigenic E. coli, Campylobacter jejuni, Salmonella, or other enteric bacterial pathogen due to poor hygiene in food preparation or inadequate water treatment. The negative stool culture directs us to another likely cause. Two of the most common parasites associated with diarrhea would be Cryptosporidium and Giardia. Giardia is relatively easy to detect in stool with transmitted light microscopy, although shedding of cysts can be intermittent and could explain the negative O&P result. But the symptoms are not typical of the foul-smelling, fatty, floating stools we rarely relish to examine for Giardia. 3 coccidian parasites could be the cause of these post-vacational sequelae: Cryptosporidium parvum, Cytoisospora belli, and Cyclospora cayetanensis. All 3 stain well with acid-fast staining techniques, which are necessary to improve the microscopic detection of these smaller and less structurally distinct organisms. An auramine stain smear positive for Cryptosporidium oocysts examined under UV illumination is a thing of beauty and could have been a likely explanation for the frequent evacuations of the septuagenarian pair. However, this infection is not treatable with sulfamethoxazole trimethoprim combination drugs, and so would not fit the pattern with the resolution which seems to be associated with treatment. Cryptosporidiosis in the immunocompetent individual should normally resolve within 7 to 10 days, and there's no suggestion in the notes that the unconstipated couple are immunosuppressed in any way. This leaves us with the 2 coccidians that could cause these symptoms and are both more common in the tropics and subtropical areas of the world. Both these agents will respond to treatment with the drugs prescribed and are transmitted through food and water where good hygiene is difficult to achieve. Cytoisospora belli infection is reported to be associated mainly with immunocompromised individuals and is considered to be rare. Cyclospora cayetanensis is becoming more frequently encountered and associated with traveler's diarrhea and outbreaks particularly associated with soft fruit berries. The epidemiology of this organism suggests that humans are the main host, and perhaps the poor hygiene in camp resulted in the sharing of more than bon appetit during meals. Without more details of the acid-fast microscopy, we will place our chips on the likely cause to be Cyclospora cayetanensis. Thank you again for your wonderful podcast and stimulating case studies, David, on behalf of the University of Central Lancaster Parasitology Club.
Chris writes, "Good morning, docs. It's a beautiful 70F day here in Stony Brook, Long Island, with sunny skies all around. First, I would like to start this email by saying thank you to all 3 of the TWiPsters, as your continued persistence through this pandemic has been greatly appreciated and informative. Now for my case guess, I believe the patient has Cystoisospora belli, a protozoan parasite that lives in the intestines of people and is known to cause watery diarrhea. This parasite is typically well tolerated by most people, but is of great concern if you are immunosuppressed and has caused serious illness for many HIV/AIDS patients. This parasite is normally transmitted through the fecal-oral route and is best avoided through proper food handling and general hygiene, thorough hand washing. Attached below is a new article from a little while ago about the Peconic Bay scallop die-off that occurred earlier this year. The die-off revealed the presence of apicomplexan parasite heavily infecting the kidney of these animals, discovered by the Marine Animal Disease Laboratory at Stony Brook. The heavy infection, in addition to warm waters with low oxygen, is believed to have caused the massive die-off. Thought it would be interesting to share such local parasite news, and provides a link to an article in the Suffolk Times. Another topic of possible interest is an observation I've made over the last year concerning parasites of horseshoe crabs. Dickson likes to say that successful systems attract parasites, which I fully agree with. However, horseshoe crabs, a species that is 450 million years old, is surprisingly not host to many parasites. There is a scarcity of information on the topic, with the last papers addressing horseshoe crab parasites from the 60s. From my own personal necropsies and literature review, I've gathered that there is only one trematode species that infects these dinosaurs, a Metacercaria microphallus limuli, 2 nematodes, Monhisteria and Granthonema species, of which I've only observed one, a few protozoans not very well investigated, so probably underreported, and a handful of triclad turbellarian worms. However, these are still commonly referred to as commensalist. Just an interesting observation that such a stable host, arguably one of the most stable, has a relatively small amount of parasites community. So also I thought Dickson might enjoy the observation, as he's a fan of both parasites and has mentioned he's fascinated by horseshoe crabs. Lastly, I would like to point out that Springer recently made a few hundred textbooks free online on topics from all disciplines, so I thought the TWiP listeners might enjoy the link. I've already picked up 2 books on survival analysis. It provides a link for that. Once again, thanks for everything, and stay safe. All right, Dickson, we're back to you.
Kevin writes, Safari Guide done got the flux. What's this whole world coming to? Things just ain't the same anytime the hunter gets captured by the game. That's a 1966 song by the Marvelettes, for your listening pleasure. This case has some ironic asymmetries. As a consequence of visiting the largest herbivores and predators on Earth, our patient's intestine has become the destination of a microscopic tourists. And, apropos of nothing in particular, I'm reminded of a popular T-shirt trope from the recent past. My parents went on safari and all I got was this lousy protozoan. Well, that's an alliteration of that. This case couldn't be regarded as a rerun, despite the culprit's appearance in TWiP 180. I say this because Cyclospora is something of a newcomer to the table, having been definitively described in 1993. Some relevant tidbits follow. Cyclospora cayetanensis, a coccidian protozoan parasite. No known animal reservoir. No experimental animal model. Worldwide distribution. Another bullet point. Incubation period averages 1 week, range 2 days to 2 weeks. Fits well with our case. Another bullet point. Clinical descriptions highlight the voluminous and watery nature of the diarrhea. Another bullet point. Diarrhea can be diffuse, can last weeks to months, and may be remitting and relapsing. Source of infection is consumption of contaminated food, but waterborne and soil-borne infection also occur. Various fresh fruits and vegetables are frequent sources, berries and leafy greens, presumably due to the difficulty of cleaning these thoroughly. North American outbreaks frequently cite raspberries and cilantro. Uncommon, rare post-infectious syndromes such as Guillain-Barré and reactive arthritis. Diagnosis most reliably via nucleic acid multiplex array testing such as BioFire assay outperforms microscopy. For example, wet smears and acid-fast staining. Trimethoprim sulfamethoxazole is the antibiotic of choice, though can use Cipro in sulfa-allergic patients. Nitazoxanide has also been used. CDC Yellow Book 2020, the risk of Cyclospora is highly geographic and seasonal. The most well-known risks are in Nepal, Peru, Haiti, and Guatemala. I think I would throw in South Africa into that one. Prevention should have been the best policy, but being on safari in the middle of a water shortage has some drawbacks. Fortunately, our adventurer didn't seem to pick up any other diseases. TWiP 3 some hardest working guys in New York. How do you figure? Huh? How do you figure? Come on, Daniel, pipe up. This is your lingo.
Yeah, that's the way I would say it. How do you figure?
No, I hate to say this, but Tony Fauci has that same accent, and I don't have trouble following him at all. I don't know how much more you want me to read of this, Vincent.
I want you to go down to a terminal curiosity and read that.
Glad to do that. A terminal curiosity. Attempting to remain fashionable in plague time and trying to stray from the classics like Defoe and Camus, I read through Richard Mead's A Short Discourse concerning pestilential contagion and the methods used to prevent it in 1720. I highly recommend this short and delightful essay. Though often suffering from many 18th century erroneous medical theories, the short discourse contains many items that has a distinctly current ring to them, a passage that I found particularly relevant. Quote, leaving the place infected is the surest preservative, so the next to it is to avoid, as much as may be, the near approach to the sick or to such as have but lately recovered. For the greater security herein, it will be advisable to avoid all crowds of people. Nay, it should be in the care of the magistrate to prohibit all unnecessary assemblies, and likewise to oblige all who get over the disease to confine themselves for some time before they appear abroad. By the way, Mead also contributed some observation on our old faithful pal Sarcoptes scabiei. Oh, that's interesting. Cool, I like it. End of terminal curiosity.
All right, that's our guesses. It seems to have a theme, don't you think, gentlemen?
I feel like there's a theme.
If we get this wrong, we'll be very embarrassed.
Well, so should we let each of you make a guess?
Mm-hmm.
Okay. Dickson, sure. Would you like to go first?
No, I agree with the majority opinion in this case. The vote is Cyclospora cayetanensis for me. And it's interesting. I was there during the water shortage in South Africa on safari, and we didn't encounter any trouble whatsoever. So I'm sorry to hear that these people did. And I guess it must have depended on which company you went on safari with.
You know, I heard watery diarrhea, and I thought it would be Cyclospora, although there was another cystoisospora, I guess, is another possibility, but I didn't consider that. I thought it was Cyclospora. I thought I like this thing of it being negative on the ONP, but the acid fast ONP positive, I think, was also telling.
The drug treatment, though, I thought that
They think that's the key?
I believe. For me, it was, because up to that point, it could have been Cyclospora, a cryptosporidium, or a cystoisospora. But the drug narrowed it down to just two, and one is much more common than the other one. So.
Okay. Well, I was gonna throw in the plug for our fundraiser before I give the answer, right? So that because everyone is now listening, right? They're on the edge of their seats, like, what could it be?
Tune in next week, folks. For
No, so we're doing a fundraiser at Parasites Without Borders. So encourage everyone to go to parasiteswithoutborders.com and donate. Here we are in June of 2020. So all of the donations that are received by Parasites Without Borders in June, we will be giving for every $1 we will be receiving, we will be giving $2 up to a total of $40,000 being contributed to support FIMRIC, Foundation International Medical Relief of Children. And I think some of our listeners may be familiar with this organization. They have clinics throughout the world. Some of the stories that I tell actually come from times I've spent at the different sites. The clinic that I go back to quite a bit is in sub-Saharan Africa, in eastern Uganda. And so we're going to encourage people to help us raise money, because during COVID-19, not only are these areas suffering because of the impact of the virus, but they're also suffering because of the impact on all the tremendous organizations that help people in these areas. So we're going to encourage people to do that. And now to our exciting conclusion, our reveal. So there were several characteristics of this case that I thought were suggestive. So one of them was the duration of illness, right? This is an individual who is still having watery diarrhea 2 weeks later. So it's persisting. The other was the quantity of loose stools, which I think is pretty characteristic, 12 a day. The other hint, right, was the stool ONP being negative until the acid fast was applied. And then the other was the response to the trimethoprim sulfa. So yes, the plurality got this right. So this was Cyclospora cayetanensis. And this was confirmed by the GI PCR and the stool with the acid fast staining.
And you told us the treatment. And I think you told us also that...
Yeah, the treatment was the trimethoprim sulfa, or the co-trimoxazole. And we had a couple of questions that I think I'll answer. So what if you did nothing? If you did nothing, this diarrhea tends to go on for 2 months. So you can imagine that, right? You go to this wonderful trip, and now you have 12 stools a day for 2 months. So treatment is key. And when I've treated this, people get better pretty quickly. Usually within 24 to 48 hours, they're much better. But then you want to watch, because people will often develop a lactose intolerance, which will last for weeks. So even though you get better from the diarrhea, you want to be very gentle, very slow in the introduction of your dairy products, your milk, back into the diet.
All right, let's give away a book.
Got it.
I count 14 people who have not won a book. Let's pick a random number between 1 and 14.
Here's the drum roll.
Number 5. Here's number 5. Let's see. Scrolling.
Is that
Elise?
Wow.
Let's see. Winner. I don't know if you've won a book before, Elise.
Well, she can't say
it's been a long time.
But you're a winner in our book either way.
So if you have not and you would like us to send you a copy, TWiP at microbe.tv. Please send your address. All right. What's next? Paper. This is a Nature Communications article. A microsporidian impairs Plasmodium falciparum transmission in Anopheles arabiensis mosquitoes. Very interesting. First author, Jeremy Harrens. Last author, Stephen Sinkins. They come from the International Center of Insect Physiology and Ecology in Kenya.
ISAP. That's ISAP. Yes, indeed.
ISAP, MRC, University of Glasgow Center for Virus Research. University of Nairobi, University of the Witwatersrand in Johannesburg, the Wellcome Campus in the UK, and the Pan-African University Institute for Basic Science, Technology, and Innovation, also in Nairobi. So, as everyone knows, some mosquitoes can be infected with a symbiont like Wolbachia.
Right.
And that will prevent viruses from reproducing in them. And so there is an experiment currently ongoing in Australia and in Singapore to release Wolbachia-containing mosquitoes and see if it decreases, like dengue, for example.
I think that's interesting to say everyone knows that. I thought it was controversial. I thought, like, we all know that a lot of worms have Wolbachia, but there was, this is, I guess, the parasite.
Wolbachia is everywhere, Daniel, in invertebrates. It is absolutely everywhere.
It's in a large percentage of insects, but Aedes aegypti that transmit a lot of these viruses do not have it naturally, so they have to put it in and then release the mosquitoes.
Right.
And the key here is that Anopheles, it's not clear if they have Wolbachia at all. So they looked here for something else that might be in Anopheles species, which of course are the malaria transmitters, right? Mm-hmm. Dickson, yes.
Yes.
And they found a microsporidian. Dickson, what's a microsporidian?
It's a rather small organism that causes a lot of problems with crustaceans, particularly crabs and shrimp. And it's found throughout the world. It's got a very unusual structure inside of it. It looks almost like a nematocyst.
What is it, though?
Hydra, the coelenterate that have got the nematocysts along the sides of their tentacles. This
parasite, Dickson? It's a protozoan?
This is a protozoan, absolutely. And it's very small. It's smaller than the average size protozoan. So it's often confused for fungal spores and things like this, but it's deadly when it gets into things. And I'm surprised that Anopheles arabiensis can actually tolerate it, because it's usually pathological.
So they did a scan of geographically dispersed populations of Anopheles arabiensis in Kenya, and they found up to 0 to 9% prevalence, which is not huge, but you could re-engineer it if you wanted to and release. But it's vertically transmitted with high efficiency from mosquitoes, and it impairs transmission of Plasmodium.
That's remarkable.
Which they did by taking infected donor blood,
right?
And looked at transmission. Give the mosquitoes blood meals and look at transmission, and it really inhibited them. which is quite interesting. And then they found that it basically reduces the establishment of oocysts in the mosquito midgut and impairs colonization of the salivary glands by the sporozoites. So the key here is that it does not confer a fitness cost upon the mosquito, because as you said, good. Dickson, you said you were surprised that it didn't impair.
Yeah, I am surprised actually,
because they say unlike.
Yeah, the size, right? The size is an issue, but it's interesting in that, this is something that can be acquired through ingestion because it has this wonderful chitinous cell-walled little cyst that can be ingested, the spore that can be ingested. And that there's no fitness cost. So if you somehow are getting this into the mosquitoes, they're not going to be compromised in any way. So they're going to hopefully be able to compete with non.
Yeah, that's important because you want to establish a mosquito colony and then release them, right? That contain and if they can't compete, then that's not going to work.
As I understand it also, I think all microsporidia are intracellular parasites. There was a guy by the name of Earl Widener who was a postdoc with William Traeger while I was there, and he worked on a microsporidium that infects crabs because it had an economic aspect to it. The blue claw crab of New Orleans and the South, basically, but he ended up teaching at LSU because the crab industry down there was being threatened by these microsporidians, and they wanted to know more about them. They're beautiful organisms when you look at their ultrastructure. They have some wonderful subcellular particle distribution patterns, which I was fascinated by.
By the way, they know the species. It's called Microsporidia MB. They sequenced its ribosomal RNA, and they can do that.
Nice.
And they conclude that this Microsporidia MB is a candidate for a stable vector population replacement strategy. As an unmodified Anopheles-associated inherited endosymbiont, it provides attractive prospects for malaria control.
Wow.
I think this is going to be an important issue, right? As we're in the time of COVID, and there's been a lot of shifting of finances. There's been a lot of compromise of the activities of NGOs. How are the malaria programs going to go? How are the insect programs going to go? So I think there's going to have to be a lot of rethinking about how do we deal with this issue with malaria and the mosquito when we're going to have shifting of resources and focus.
It's going to upset the entire landscape of healthcare, I think.
I like the mechanism that they were suggesting here too. The idea that the microsporidia MB infection was actually modulating the host genes. Specifically ones involved in digestion, immunity, and of course transferrin, which I know has been discussed on TWiP, our big sister podcast.
Why wouldn't that then exact a cost for having a parasite there?
Very good question.
Yeah, I mean, it's interesting because you'd think maybe the transferrin, is a way of limiting the iron, limiting the impact of the plasmodium. But maybe the insects are not as reliant on, higher levels of iron. It's hard to know.
They do say there are reports of Wolbachia in Anopheline mosquitoes and some evidence for plasmodium protective phenotypes. But they say they may have low efficiency of vertical transmission, et cetera. So that's why they looked for something else and they found this. So hopefully
it's too bad there isn't a fitness price to pay for it, because it would have selected for the resistant strains of these mosquitoes that then could never transmit malaria afterwards.
I guess the hope would be you could put something in like a microsporidium that might even have a fitness advantage, right? So somehow you want your because we think there's some degree of a fitness cost to having the plasmodium in the mosquitoes, but not significant enough, unfortunately.
All right, Dickson, do you want to
I do.
Submit this hero?
I would like to, because I didn't have one selected per se. I was scanning the horizon of the past, trying to decide on which of the people that have emerged over the last 40 years as leaders in this field. But Someone has done that for me, thank goodness. His name is Dr. Martin Nielsen. He's a veterinarian in the Department of Veterinary Sciences at the University of Kentucky in Lexington, and he suggests somebody that I think is a great addition to our stable of heroes because we don't have one of these yet, but I think we will now. So he suggests that we elevate or recognize, better to put recognize, Dr. Eugene T. Lyons. He writes, and he described this gentleman for us. Eugene T. Lyons is a legend in equine and sea mammal parasitology. He was the first to describe a lactogenic route of transmission for a nematode parasite, which was considered unlikely, if not impossible, at the time. He first unraveled the life cycle of the hookworm of northern fur seals, Uncinnaria leucasi, and demonstrated how larvae were passed from mother seal to her pups through the colostrum. Then he went on to demonstrate lactogenic transmission of Strongyloides westeri larvae in horses. He worked here at the University of Kentucky for 56 years and has published over 300 peer-reviewed scientific papers. His contributions also include works on life cycles of other important parasites in horses, as well as pioneering drug discovery and testing. He was instrumental in documenting drug resistance in important equine parasites. He worked 7 days a week, year-round, and only vacation he ever took was to participate in field trips studying parasites of his beloved seals and sea lions. His day would start at 6 a.m. in the morning, and he would continue until evening time every single day. I had the good fortune to work with him and learn from him for several years before his passing in December of 2017. He was humble but generous with his knowledge, and he enjoyed interaction with young people, teaching future generations of parasitologists. You can read more about him in the memorial piece attached that I wrote for him, but perhaps we'll post that in the show notes, that I wrote for him and his technician, Sharon Tolliver, who were inseparable over 45 years of working together, and she passed away just a couple of months before. I also enclosed his publication list and a few pictures of him. That's a very, very good contribution. And you know what? I think we should throw this open to the listening public. If they have similar people that they want to have recognized, we would be glad to consider them for our heroes in parasitology. So thank you for this contribution.
All right. And now I believe it is time to see if Daniel has another. I know he's got at least one more case study.
You know, that's funny. I have several.
Strange as that may
and you know, I was reminded as we were reading, I did promise that I would give the second half that we would discuss the wife. So for everyone listening, here's the story. So we have a 69-year-old woman who comes in reporting, now this is several months later, several months of abdominal symptoms that started while she was on safari in South Africa with her husband. She reports that when she and her husband arrived in South Africa, there was a severe water shortage. And even though it was supposed to be a fancy trip, there was limited water for washing, and they were using hand sanitizer to clean their hands. She, too, about 5 days after arrival, both she and her husband became ill with diarrhea. We now know the husband had Cyclospora cayetanensis. But her symptoms improved somewhat, but not completely. But when her husband was diagnosed, she was told, not by me, someone else, that she needed to be tested as well. So she's under the care of a different clinician at this point before she comes and sees me. And her stool came back positive for blastocystis hominis and endolimax nana. She is then subjected to a lot of treatment. She was first treated with metronidazole, then tinidazole, then paromomycin, and then nitazoxanide, and then Bactrim. She now comes to see me, saying that she is afraid to see her grandchildren for fear that she will infect them. She currently reports some abdominal discomfort, mucosy stools, and just not being herself since this trip. Her past medical, past surgical is all unremarkable. She's not allergic to anything, despite, exposing her to everything. Nothing runs in the family. And she is sent for repeat lab testing, including a stool O&P. And all the testing comes back completely normal, negative. This includes a GI PCR as well. So what do we do? And so that's what I'm going to leave
She's
people
HIV
with.
negative, right?
Right.
What does she do? What does she do? Say she is HIV negative.
And she was with her husband on this entire trip, right?
Yes.
That's remarkable. Very remarkable. I'm surprised that they didn't do stool exams after each treatment to make sure that she was still infected with whatever they thought she had before giving the next drug. That would have been a logical approach. But that wasn't the case. They threw the kitchen sink at this woman, is what you're saying.
They kept treating her, trying to resolve this abdominal discomfort. So she went through a few months of repeated treatment, and now she's seeing us, and we're gonna address this hopefully.
Sorry, was she examined for other non-infectious diseases that might have caused similar symptoms?
No, so there was no endoscopies, no other workup. It was just a one drug after the other.
And didn't have anything before she went on safari.
No, she was in her usual good state
Apparently
of health
they
with
both
no medical.
ate different things. At least that's what it suggests.
Not necessarily, Dickson. Not necessarily.
That's true.
People
you're right.
could be
You're right.
Differentially susceptible. And who
You're
knows,
absolutely right. You're absolutely right.
I mean, I'm thinking the same thing that, maybe she went off and ate one night, but no, it could be just you eat the same thing and one person gets one and one person.
But you know what,
gets the
Vincent?
other, so.
When I was on safari with Marlene, it sounds kind of uppity. When I was out looking at wild animals,
When you were on your fancy safari,
they served most of it as a buffet style, so that you had a choice of lots of different things to eat. And Marlene and I never picked the same things, so we would have eaten different things all the way through our safaris, I think. So that's possible here too. I mean, you don't have to eat exactly what everybody else is eating.
Okay, that's interesting.
Yeah.
I didn't know that because I've never been on safari.
But you have been to a buffet dinner, I bet.
I think maybe I did once. Yeah. Yes, of course, a buffet dinner. But of course, even if you sit down at a restaurant, you can have a different dish.
And Wendy's always has the buffet salads, and everybody comes back with something different.
All right, that is TWiP 184. Folks, listeners, you got a month to solve this case. Start working on it. There you go. If you like what we do this week, this month, why don't you go to parasiteswithoutborders.com and donate there, and they will match your donation twofold. To what's the name of the organization, Daniel?
Parasiteswithoutborders.com is supporting FIMRIC.org.
FIMRIC. So go to parasiteswithoutborders.com. And if you have any questions or if you have a guest for this month's case, TWiP at microbe.tv. Daniel Griffin is at Columbia University Irving Medical Center, parasiteswithoutborders.com. Thank you, Daniel.
Pleasure as always.
Dixon Despommier is at Racaniello.org and thelivingriver.org. Thank you, Dickson.
It was fantastic. Thank you.
Over on TWiV, last week I said you're livingriver.com, and someone wrote in and corrected it. They said,
"Oh, really?
It's thelivingriver.org," and he said, iOwn.com if you want to buy it.
Oh, that's great.
Oh, boy.
That is funny.
Oh, boy. I'm Vincent Racaniello. You can find me at virology.blog. I want to thank Ronald Jenkins for the music and ASM for their support. You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon. Another TWiP is parasitic.